Hi all, so welcome to this session on cell signaling. Today we're going to be talking about why cell signaling is really important. We're going to talk about what the basic types of cell signaling are, the three main types. We'll also look at the cascade effect and then some specific key examples of cell signaling. So, cell signaling. It's really important that cells can communicate with one another. In a multicellular organism, of course, this is absolutely critical to enable the organism to function effectively. We've already talked about how single-celled organisms can form quorums, so bacterial quorums, but this is so much more important in a multicellular organism because different parts of the body need to be able to communicate with one another, talk about what's going on, and direct other parts of the body to carry out the correct processes. So how do cells communicate? Well, you might think with a cell phone, but well, clearly not. Instead, they're going to be using signal molecules. The first type of communication between cells is through direct contact. Direct contact is just like passing a note to your neighbor. It's the two cells right up next to each other, and the message is passed directly between them. The plasma membranes of the cells are adjacent to one another, so it's all very straightforward. Direct contact, so you have gap junctions between animal cells and these things called plasmodesmata between plant cells. And in both of these cases, what's going on is the cell's cytosol is actually able to move between the cells. Molecules are able to pass between the cells without crossing the cell membrane specifically. And so that obviously speeds up communication and enables cytoplasmic streaming between the two. So that's what direct contact. Direct contact we've also mentioned in terms of antigen presentation. You may remember that dendritic cells and other phagocytes, when they break down bacteria or other pathogens, they will then put part of that bacteria, part of that what's called the antigen, which is the surface structures, on their own surface. They present it to the helper T cells. This enables the helper T cells to identify and the foreign body, and it activates the helper T cells, which will then further the immune response. So that's a specific example of direct contact. Paracrine signaling is between local cells. The cells aren't necessarily adjacent to each other. It's a bit like throwing a paper airplane to your friend. The cells aren't right next to each other, but the signal molecules are just past a short distance. So they'll be released into the interstitial fluid between the cells, and it's a local message. This is paracrine. Think of parallel being next to one another. Paracrine means to be close to one another. That's a local mediator going to a receptor on the surface of a target cell. There is a form of signaling called autocrine, which is when a cell releases a signal molecule for itself. Some T cells do this to promote their own growth in the immune response. The endocrine system and the endocrine messages are ones that are being sent. They're like broadcast messages, just like posting something on Facebook, where you're sending it out to the whole body. Now, it may just have specific, in fact, it normally will have just specific target cells that are trying to reach, but the signal is being sent all over the place. So hormones are good examples of this. So phosphorylation is something that we're going to be talking about a little bit, and it's important to understand that phosphorylation acts like a switch. If proteins are not phosphorylated, if enzymes in the cells are not phosphorylated, typically that means they're inactivated, and so that they aren't carrying out their normal function. So for example, here we have part of the cell cycle an important stage. This RB protein here is preventing this E2F transcription factor from reading the DNA. You'll learn more about how transcription is controlled in a later session. But it's important to understand here, this is not allowing this to work. So transcription, the genes specifically here needed for S phase, are not working. However, if this becomes phosphorylated, then the RB protein moves off the transcription factor, allowing it to carry out transcription and produce the proteins needed for S phase. CDK, which we've mentioned before, cyclin-dependent kinases, in conjunction with cyclin, work to phosphorylate this protein. Anything that's a protein kinase, think kinetic, means getting things done, movement. Protein kinases are enzymes that phosphorylate other molecules within the cell, enabling them to be switched on effectively. Phosphatases do the reverse. And so all of these, it's very clever, these are effectively molecular switches, can be switched back off again. Another term that we're going to be using that you need is ligands. Ligands are any of these signal molecules. Okay, so it might be a hormone, it might be one of these local signal molecules. It's the primary messenger between cells that is detected by the receptors in the cell surface membrane. And that's going to trigger a response. First of all, we're going to have a receptor binding to it. Then we'll have the signal transduction, secondary messages within the cytosol. We'll talk more about those in a moment. That's going to lead to either cellular responses, changes in the cell's gene expression, or possibly both of them. So there's lots of different types of these receptors on the surface of cells. Some of them are channel linked, and so when the signal molecule binds to the receptor, this opens up a channel. Now that can be directly, or it could be secondary, attached by a cascade of events. But in this case, you can see in example A here, this is a direct channel-linked receptor. The signal binds to it, it opens the channel, and ions can flow across the membrane. We'll see specific examples of this when we look at the nervous system in detail. Example B here is an enzyme-linked receptor. So the signal binds on, and that activates this enzyme to operate on the inside of the cell. So this will typically be something like a protein kinase that we mentioned before. It'll phosphorylate something, creating a chain of events within the cell. A nice example of this is insulin. Here we have a protein kinase. Insulin, this hormone, remember, that is released when there is an excess of blood sugar, is detected by the insulin receptors on the surface membranes of the liver cells and muscle cells. The protein kinase phosphorylates this insulin receptor response protein on the inside of the cell membrane. In the cytosol, the phosphorylated insulin response proteins activate glycogen synthase, which does what its name suggests. It produces glycogen out of glucose, thereby lowering the amount of sugar that's present. So this is the mechanism by which that detection takes place. This is a typical cell signaling event, and one that the AP could ask you about. Make sure you've got that detail down. Membrane receptors, there's two other types of receptor that you need to know about. There are the G-linked or G-protein coupled receptors. These are receptors that have a separate protein part on the inside called the G protein that becomes activated when the signal binds to it, when the ligand binds to it. Talk more about those in a moment. There are also intracellular receptors. These are on the inside, not attached to the membrane. For a signaling molecule to activate these, they need to be able to pass through the phospholipid bilayer. So typically they're going to be non-polar. Steroids are good examples of this. Here's a nice example of this one is testosterone. Testosterone can pass straight through the cell membrane and activate receptors inside the cell that will turn on various transcription factors. There's also second messengers or secondary messengers within the cell. These are important for relaying signals from the receptors on the cell surface to target molecules inside the cell. That could be in the cytoplasm or the nucleus. These are the second messengers. So you had your primary messenger was, say, the hormone, the ligand that came to the external surface of the cell. Now that activates secondary messengers. Two common secondary messengers are cyclic AMP and calcium. So we've got a little video about cyclic AMP for you to watch here.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP from ATP within the cell. The cyclic AMP then binds to and activates a target protein, such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated.
And so that last point there is really key, that it really depends on the cell, like what is happening, what is that specific cell, that what is your response going to be. And we'll talk more about the G protein activation in just a moment. One other thing that you need to understand is the cascade effect. You imagine that part of your body has sent out a hormone. Only a very, very, very tiny amount of that hormone is going to reach the target cell. And so if that was the only one working, say switching on a transcription factor, very little of the desired effect will come about. Instead, what you need is a cascade event. That signal molecule setting off a chain of events that builds and builds and builds until you get a really large response. So in this particular instance here, the receptor is activating this relay molecule, protein kinase 1, causing protein kinase 2 to be activated, causing protein kinase 3, etc., until we reach our active protein that we're desiring. This is a bit like having a tiny, tiny domino that sets off a chain of much bigger events. We have our initial signal molecule that's individual perhaps, being detected by the receptor, and then the activator, which these kinases, these protein kinases again, activating more and more and more proteins until we reach the response proteins, a cascade starting small and getting really big until we reach a significant cellular response. So I want to talk now about the G protein and cyclic AMP or CAMP signaling pathway. This pathway is a bit of a classic. It comes up a lot and it's one that you should know about. The ligand, first of all, binds to the receptor on the cell surface. I would copy these diagrams as you go through. That receptor is linked to a G protein. Okay, this is where the G protein or G-linked receptor comes from. And that has 3 different subunits on it. The binding of the ligand to the receptor causes the G protein subunits to dissociate, so they split up. And the alpha subunit is the critical one here. The alpha subunit splits away from the other 2, and the GDP that is on its surface is swapped for a GTP. Okay, think ATP, sounding familiar, GTP, similar energy carrying molecule, okay, triphosphate. This alpha subunit moves over to the adenylate cyclase. Now, adenylate cyclase, that's going to be a tough one to remember. I just remember going to Denny's at 1 o'clock in the afternoon, right? It was a late Denny or a Denny late. And cyclase, it's going to be producing that cyclic AMP or CAMP. The adenylate cyclase is activated by this alpha subunit. and it converts ATP to cyclic AMP. Hang on a second. Now we've covered ADP before, right? And ATP, adenosine triphosphate, broke down into adenosine diphosphate. But now we're producing cyclic AMP, adenosine monophosphate. In other words, you're losing 2 phosphate groups and making a slight adjustment to call it so that it's cyclic. So this is the CAMP that is produced, and this is that secondary signal molecule. The CAMP then activates protein kinases within the cell. And those protein kinases can go on to do all kinds of different things. So, key steps here. The ligand binds to the receptor, which causes the G protein on the inside of the cell to dissociate. The alpha subunit activates the adenylate cyclase, and the adenylate cyclase produces this cyclic AMP or CAMP. The CAMP, the secondary molecule, is activating protein kinases. And remember, the protein kinases are what gets stuff done in the cell. So, for example, the protein kinase might open an ion channel. It might phosphorylate other proteins. Nice example is, say, a growth factor could be detected by a receptor. We get a phosphorylation cascade that results in an inactive transcription factor being turned into an active transcription factor. And the response is the transcription of a particular gene, producing the mRNA and protein that is required by that growth factor. So you can think of this as like a computing event. Okay, we've got our input, we have our output. And the calculation that takes place in between is determined by the specific receptors and the specific cascade that takes place of events. So one critical thing to understand here is this really applies to a lot of medicine and disease. Many diseases involve signal pathways either malfunctioning or being exploited by a pathogen. So an example of this is the cholera toxin. The cholera toxin is passed into the cell by endocytosis. It induces endocytosis to bring itself into the cell. It then causes one of these G protein pathways to be activated, producing a large amount of CAMP, cyclic AMP, and that in turn causes a large amount of chloride ions to be pumped into the lumen of the digestive system. Pumping chloride ions in causes sodium, positively charged, to follow as well, and water by osmosis. This causes a huge amount of water to move into the intestinal tract, the lumen of the intestine, causing severe diarrhea and allowing the cholera pathogen, the bacterium, to spread itself through the fecal-oral route. So this way, the cholera bacteria releasing the cholera toxin is actually exploiting a pre-existing pathway within human intestinal cells. So, to sum up, you should be able to label this diagram. If you can't do that, make sure you watch this YouTube below. It goes over this stuff again. I'll put the link in the S drive. Thanks for watching, guys.